So this is a video for people who are still confused on how to do this worksheet. So again, we have DNA. We need our DNA to make our cells and the proteins within our body. So how does it do that process? Well, it does it through transcription and translation. So it says here transcribe and translate. Transcribe is our first part, which is going to just make a copy, and that copy is mRNA. Okay, so that is what we see right here. That is that copy, and that copy is extremely important in that it is the instructions. So each 3 bases is what we call a codon, and that is the code in which we will use to make our protein. So this was again copied during transcription, and translation is when you break down this code to figure out what your amino acid sequence is that then makes that protein. So number 1 is determine the sequence of amino acids that will result from translation. So now we're translating it into the amino acids. So again, we look at every 3 base. So U is our first one, U, C, U. Base number 1, base number 2, base number 3. So number 1 is our first nucleotide, and that is going to then be in this row. Our second nucleotide is C, so really we've narrowed it down to this box right here. So I don't even have to go further because there's only one amino acid for either of these sequence. So my first is SER. My second amino acid is C, and I'll change my color for this. It is CGU. So C, it's going to be in this column here. G, so again my second base, so it's going to be in this box. So again, I don't even need to look at my third base, but again, I'm trying to match this code with the code up here. So I'm just matching. So then it is ARG. If you have any further questions on this, please see me. But again, all it is matching the code with the code within the boxes. And this is just the first, second, and third is just meant to help you on how to figure it out. Instead of looking through every single code in the box. Okay, number 2. Number 2 is going to be about the anticodon. So I'm going to clean this up a little. So the anticodon is what is found on our tRNAs. So you have all these tRNAs floating around. They have their own code, and they're going to match up with the code on the mRNA. So if you think about it, U goes with A, C goes with G, and U goes with A. So you have this tRNA hanging around. It has an amino acid on it, and it's just going to match up with our codon on our mRNA. And again, we already saw that that amino acid is our SER. So that's the amino acid hanging on our tRNA. The tRNA is going to just transfer that amino acid onto the chain. So it just says determine the anticodon. Well, the anticodon is the opposite of our codon. And that's how I got my A, G, A. So I use this key to figure out what base is going to go with what. That is for our RNA. And then I'll just continue it on. So from this one, C goes with G, G goes with C, and U goes with A. Again, using this down here. Then G goes with C, A goes with U, and U. And I continue that on. I'm going to leave that blank for you. If you have any questions, we'll do those together. Number 3 is then asking critical thinking, recognizing patterns, determine the sequence of nucleotides in the segment of DNA from which this mRNA strand was transcribed. So again, transcribed is just making a copy of our DNA. So this is just asking, what would the DNA read? because we made a copy. This is our copy. What's the other side of the strand? So we're going to use those base pair rules. But instead of having a U, since we're talking about DNA, I'm going to now have my T. Thymine exists for DNA. Uracil exists for RNA. But again, DNA, so I'm only thinking about the T. So now let's start making our copy. So U goes with what letter? Well, U goes with A. That A still exists in our DNA. It still exists. C goes with G. A goes, or U goes with A. And I continue on. C goes with G. C. A. G goes with C. A goes with, now that we're talking about our DNA, it's not uracil, it is T. And I'll continue that on. Number 4 then is asking, determine the sequence of nucleotides in the segment of DNA that complements the DNA segment that's described in item 3. So basically that is asking me to tell me, or you're going to tell me what the other strand of the DNA is. So what complements these? What are the base pairs? So A again goes with T, G goes with C, A goes with T. And I continue on. If you notice something, it's almost the same as up here, except for whenever there's a U, I replace that with a T. And I continue that on. Now this is supposed to be active reading, so you're supposed to read this passage below and then answer the questions below here. So like DNA, ribonucleic acid, RNA, is a nucleic acid, a molecule made of nucleotides linked together. RNA differs from DNA in 3 ways. I'm going to highlight that instead. In 3 ways. Or not. First, RNA consists of a single strand of nucleotides instead of 2 strands. And again, if you think about transcription is when it copies the DNA. One strand is smaller, it's going to fit through that nuclear pore and go out into the cytoplasm much more easily. Second, so back to the reading. RNA nucleotides contain the 5-carbon sugar ribose rather than the sugar deoxyribose found in DNA, which deoxyribose is why we have our name DNA, deoxyribose nucleic acid. So it's named after the sugar. Okay, back to the reading. And third, RNA has a nitrogenous base called uracil, abbreviated U, instead of the base thymine, found in DNA. No thymine bases are found in RNA. Like thymine, uracil is a complementary to adenine whenever RNA base pairs with another nucleic acid. Answer the questions below. In the space below, write a D if the statement is true for DNA. Write an R if the statement is true for RNA. Write a B if the statement is true for both DNA and RNA. So you're going to have to here put a D, B, or R. And then the last thing, an analogy is a comparison. In the space provided, write the letter of the term or phrase that best completes the analogy. RNA is to U as DNA is to what would fit best? C, G, T, and A. Good luck. If you have any questions, please see me after school, 2nd hour, 5th hour. I have last lunch. Or see a tutor during PLT.